Okay, so I flipped my camera around a little bit. That's the first time I tried one of doing these on the board, and I did need to see where my spacing was. I will also attach a JPEG of the first part of this so that you can see, but now you can see where we're working it up. So again, other info and details, you're going to be adding more here. We started with vocabulary and terms, structure, or the diagrams for these, and then an output column. So as we get to the nucleotide and triplet, and here's our triplet, then when we left, we were taking a look at how right at about what spot in your vocabulary terms that RNA comes and makes some changes. RNA is kind of the molecule that can roam in and out of the nucleus, which is where DNA lives all the time, it never leaves. Nucleic acid, it makes some edits and translations and then moves it out to where it actually gets produced. So once it makes its edits, then we call that triplet a codon. It's an edited triplet. And codons then, which are a 3-letter code, as you can see in that chart on page 41, produce 1 amino acid. Now, as you take a look at that, you will see that each amino acid, and there are only just over 20 of them, has several different ways that they can be spelled. And this is one of those evolutionary end zones that makes it actually really wonderful, because it means that small mutations that occur constantly during cell division as you read through the end of 3 into 4 and 5 sometimes really actually don't change the output for the organism, so that you're not always strongly impacted. So in order to keep this a little bit tidier for you, because you already have your part that you charted down, I'm going to take out a little of this so that it stays pretty visually clear here, and then over. I'm going to erase this now because you don't need this part. So you have your 4 letters for DNA bases, but now take a look at that chart on page 41, and you're going to want to also over here put RNA bases. The letters, what are they? The letters for them, and then the terms for them. Now you'll see in that chart that everything under that column at the top that says DNA bases are only using the letters A, T, C, G. And if you take a look underneath the one that says RNA codons, do you see a G? Yes, you do. It also includes guanine. Do you see adenine? Yes, right? You see an A in those 3-letter codons. Like one right at the top under alanine, you see that there is some on the RNA. Do you see C, cytosine? Yes. Do you see T in the RNA column? No. There is no thymine in the codons. The substitution is for uracil, and so this one is one of those really, yay, kind of easier to remember things. RNA codons are guac. There you go. So there isn't a good one that I could think of for this one. So DNA, the one that's unique to DNA is thymine, the one that's unique to RNA is uracil, the U. So 1 amino acid. Now, 1 amino acid isn't enough to make code. It just makes 1 amino acid. As you're going through your reading, though, it starts to, as you get it back, not out of the chart there, table 3.1, but into the text part that starts with at the end of 39 and goes over to the top of 40, and then the bottom of 41 over to 42 there, it says that the end zone, the big result of genetic code, is to produce a polypeptide chain. And poly means multiple or many. So obviously this needs to get grouped up into a larger group so that these get clustered and these come into a multiple amino acid chain or group. So I'm going to switch colors here. Let me find a good one. They're all good, right? I just have my favorites. All right, so we're going to go down here. Now, for triplets. Triplets don't group up in groups of 3. So what you want to do on your chart in the middle for triplets, and I used purple for triplets, is just bracket out several groups of 3 here. It doesn't really matter how many, can be lots. Now, with the green, or whatever color you're using next, I'm going to bracket all these together to indicate that what's happening here for this particular term is a grouping of triplets. And you'll see it over here, so then it'll be over this. Now we're probably finally getting to vocabulary that you have noticed before in conversation or media, that sort of thing, right? So, and I want to make sure that I've got a little bit more. My things are rolling away here as I go through some other things. So those codons in larger groups, they make the groups of amino acids. So this is also going to come down here into something called polypeptide chains. So it's going to be multiple amino acids, multiple triplets. Now here, for this one was 3. So, but down here, what's going to happen is the triplets group up in variable quantities. It depends. It depends on what organism and what function needs to be carried out. So our terms for these groups of multiple triplets, or on this side, variable clusters of triplets, is finally a word that you use all the time, which is protein. Is it? No. Protein goes over here, right? That's why charting these out gets better, because when you answer multiple choice questions, it doesn't work quite as well. So the real term here, the vocab term for just the DNA structure, is gene. That's what a gene is, you all. It's a group of multiple triplets. So here we've got it here. These all get redone into the codons. So multiple amino acids, or a polypeptide chain, equals 1. Now we've got it, protein, right? Because protein is a output. It's a biochemical. Now this part sometimes for students is a little bit more confusing, because we use the term protein in a singular form all the time when we're taught, because people say you need protein in your diet, or this is lacking in protein. However, in biology, we know that there are many, many different types or forms of those amino acid chains that produce protein. So I'm going to erase this right now because you've already got it on yours. We have a little bit more room here. We can add a couple things about genes. Firstly, for humans, right now, we know that genetic code, as we can discover it currently, and we're learning new things about it all the time, can create over 90,000 proteins. So there isn't just one type. We also know that that genetic code that produces proteins sometimes links up to protein chains in order to make the output. And one of the great examples you have in your reading at the bottom of page 41, figure 3.7, is hemoglobin. Hemoglobin protein is actually the biochemical item that makes your red blood cells. So it takes 2 chains of those to chain up. So gene. Notice also this way, genes are not separate from the rest of the chain. They're not a separate particle, molecule, or entity. A gene is an area on the DNA molecule. So that makes a gene. The term gene was invented 50 to 60 years before we actually knew where, how it was coded, or how to discreetly find more or less where the edges of this was in code. So it was a placeholder term. If you hear someone today saying genes and anything else that has to do with coding, then they're probably not, the reason I didn't go ahead further with that, I didn't want to add any new terms yet that we haven't talked about. They're probably not somebody that really understands exactly what they're talking about, but they do have an idea that genes have an output. So genes. So sequences of codons form proteins, and those will have genes. Now, humans have right now an estimated, for these, 20 to 25, it kind of depends on whose literature you're looking at, 20 to 25,000 gene sequences that we can identify, 20 to 25,000. So, right, so because they cluster together, like recipes, you have a few ingredients, but you can make more dishes out of it. That's what genes are working on. So we've got, we started with the nucleotide, then we looked at triplets, and now we're up to the term gene for you. Okay, so we're still not quite done, but we're really close to it. Let me see if there's anything else, vocabulary word, that I want to make sure that we've got for this one. We still haven't hit the big term that you're familiar with, but we only have one more level that we need to do real quickly. And then again, there'll be some explanation and examples as we meet together. Now, genes then are going to be these groupings on the molecule itself. What haven't I used yet that I have a color for? Not attractive, but you should be able to see it. For the brown one. So go ahead on yours and chart out some other bigger groups in green. Whoops, already making a mistake on mine, right? Because your sequence on the DNA molecule is going to make lots of gene sequences, but all of them together, I'm going to switch to my black pen because that brown one didn't seem like it was very strong, is going to go all the way from the top to the bottom, all of them together. So genes, we're going to drop down to the next vocabulary word, times variable quantities equal the structural strand. That's also a term that you probably use and you've certainly heard, and that is chromosome. So a chromosome is a sequence of genes. A gene is a series of triplets. A triplet is 3 nucleotides, and the nucleotides are a sugar, a phosphate, and a base. So we've got far more of these than these, right? Now in humans, how many chromosomes do we have? The total number of chromosomes in humans is 46. We have 46 of these coding strands. What happens when we go over here and drop this down, right? Because we have a nucleotide doesn't make anything on its own. A triplet turned into a codon makes 1 amino acid. Chains of amino acids make 1 protein. So what all of it together makes is multiple, you got it, proteins. So what is genetic code? Genetic code is a series of proteins. Is your body making all of the proteins it can all at the same time? No, it's not, right? Right now, you're fully an adult, so your body doesn't need to be, for instance, still growing your brain, or still creating the growth patterns and eruption of your adult teeth. That's done. That protein code got triggered earlier on, did its job, and is now turned off. for the rest of your life. So unfortunately, if we lose a tooth, we don't get to do another one. Same thing with your heart. Your heart is finished growing. And so if it's damaged, it doesn't have any more code that tells it to repair or grow more heart muscle, which is why we've got to have lots of other ways to help people out with that one. Okay, so nucleotide down to chromosome. So let's stop right here for a moment, and you can take a look at that and work through that part of the reading. Let me push my